Hello everyone, I'm Tanny Sue.
And I'm Yitty, and we're going to be talking about neural crest cells and skin pigmentation. To understand skin pigmentation, we first have to explain neural crest cells and their regionalization. And so we'll first discuss the importance of neural crest cells and the mechanisms involved. Then we will talk about who they were discovered by and how. Afterwards, we will examine the four regions that the neural crest cells migrate to and the relation to skin pigmentation. Finally, we will investigate an abnormality caused by neural crest cells called melanoma.
Have you ever wondered how your jaw became your jaw, your face became your face, or even your skull became your skull? All of these are formed from the neural crest cells. So what are the neural crest cells?
Neural crest cells are a cluster of temporary cells that are used to make other cell types. They are derived from the ectoderm, and more specifically, they are found on the margins of neural tubes. Neural crest cells are only found in vertebrates and are usually referred to as the fourth germ layer. This is because of their significant contribution to embryonic development and the large number of cell types that are formed from this embryonic region. In the figure, the localization of neural crest cells is shown in green. The three images show an open neural plate, a closing neural fold, and finally, the migration of neural crest cells as a result of the formation of a new cell type. Neural crest cells migrate to many different locations and then they differentiate. The migration is organized by the distribution of repellent and attractive signals. The different cell types produced by the neural crest range from neurons and Schwann cells in the peripheral nervous system to the adrenal medulla in the endocrine system. They are also responsible for smooth muscle, bones, and cartilage. The discovery of neural crest cells occurred in 1868 by the Swiss embryologist Wilhelm His. He was examining a chick embryo when he noticed a group of unknown cells that formed a thin band between the neural tube of the developing chick and the fetal epidermal ectoderm. He found that this band of cells was the reason for spinal and cranial ganglia. As a result of this, he gave this band the German name of Zwischenstrang, which means the intermediate cord. The names of these cells were eventually changed to neural crest based on their location by the neural tube. As mentioned before, the neural crest cells migrate and differentiate. There are four main regions that this occurs in: the cranial, cardiac, vagal and sacral, and trunk. There are some areas where these different regions overlap, but still remain highly restricted. The cranial neural crest migrates into the pharyngeal arches to form the bones and cartilage of the face and neck. It also produces pigment and cranial nerves. The cardiac neural crest cells arise from the neural crest by specific somites and are critical in making the division between the aorta and the pulmonary artery. The vagal and sacral neural crests form the parasympathetic nerves of the gut. And finally, the trunk neural crest makes up sympathetic neurons and the medulla portion of the adrenal gland. In the next few slides, we will explain each of these neural crest regions in more detail. Cranial neural crest cells migrate to produce the craniofacial mesenchyme, which forms into the cartilage and bone of the face and neck, along with the cranial nerves. These cells also enter the pharyngeal arches. The pharyngeal arches are pouches located in the head and neck region in which the cranial neural crest cells give rise to thymic cells and bones of the middle ear and jaw. Cranial cells can migrate in one of three important streams. First, the neural crest cells from midbrain and rhombomeres 1 and 2 located in the hindbrain migrate to the first pharyngeal arch, forming jawbones along with the incus and malleus of the middle ear. Some go on to differentiate into the neurons of the trigeminal ganglion, which innervate the teeth and jaw, while others contribute to the ciliary ganglion of the eye. These neural crest cells are pulled by the expanding epidermis to generate the front nasal process in which the bones form into the forehead and the middle nose. Cranial neural crest cell structures much of the facial skeleton. The second is that neural crest cells from rhombomer 4 populate the second pharyngeal arch, forming the upper part of the hyoid cartilage of the neck and the middle ear. These cells contribute to the neurons of facial nerves. The hyoid cartilage will ossify into the bone in the neck that attaches muscles of the larynx and tongue. The third is that neural crest cells from rhombomeres 6 to 8 migrate in the lower pharyngeal pouches to form the lower part of the hyoid cartilage, which contributing cells to the thymus, parathyroid, and thyroid glands. These neural crest cells enter the developing heart where they aid in constructing the outflow tracts.
The vagal and sacral neural crest cells generate the parasympathetic ganglia of the gut and control the intestinal peristalsis. The vagal neural crest, which is in the neck area, overlaps the cranial trunk crest boundary. The vagal neural crest lies opposite cheek somites 1 through 7, while the sacral neural crest lies posterior to somite 28. Failure of migration of neural crest cells from the region to the colon results in absence of enteric ganglia and lack of peristaltic movement in the bowels.
Cardiac neural crest is located right in the middle of the cranial and trunk neural crest, extending from the first to third somites. Cardiac neural crest cells develop into melanocytes, neurons, cartilage, and connective tissue. These cells produce the entire muscular connective tissue of large arteries from the heart. The caudal region of the cranial neural crest is considered a part of the cardiac neural crest because cells in this region generate the endothelium of the aortic arch arteries and the septum between the aorta and pulmonary artery. Cardiac crest cells enter the pharyngeal arches 3, 4, and 6 to become portions of the neck, such as the thyroid, parathyroid, and thymus glands. In the thymus, cells deriving from the neural crest play an important role in regulating departure of T cells from the thymus and into circulation. In mice, cardiac neural crest cells actually express the transcription factor PAX3. Mutations of PAX3 is due to fewer cardiac neural crest cells, thus leading to truncus arteriosus and defects in the thymus, thyroid, and parathyroid glands. These congenital heart problems in humans and mice could be linked to defects in the migration of neural crest cells.
Here we have the trunk neural crest cells that can migrate in 2 ways. First, the dorsolateral migratory pathway of neural crest cells allow the precursors of melanocytes to move through the ectoderm towards the ventral midline of the belly. The ventral pathway takes the trunk neural crest cells ventrolaterally through the anterior half of each skeletome. Skeletomes are blocks of mesodermal cells deriving from somites that will differentiate into the vertebral cartilage of the spine. The sensory neurons are the remaining trunk neural crest cells in the skeletome from the dorsal root ganglia. Cells continuing to travel more ventrally from the sympathetic ganglia, adrenal medulla, and the nerve cluster around the aorta. The neural crest cell migration is controlled by the extracellular matrices surrounding the neural tube. The initiation of the epithelial mesenchymal transition, or EMT, is caused by the activation of Wnt genes, by BMPs. BMPs are controlled by noggin produced in the somites. When somites stop producing noggin, BMPs function to activate EMT and dorsal neural tube cells. Wnt and BMP signals promote expression of proteins from snail, foxD, and roe families, destined to become immigrating N neural crest cells. Row B is involved in establishing cytoskeletal conditions for migration by promoting actin polymerization into microfilaments, the microfilament attachment to the membrane. The crest cells cannot leave the neural tube if they are tightly knitted with one another. Cells are connected by snail proteins to activate factors that associate the cadherin that bind these cells together. Another factor in the initiation of neural crest cell migration is the loss of N-cadherin that link the cells together. Located at the surface of neural crest cells, N-cadherin is downregulated. Melinoblasts emerge as pigment cells from the neural crest cell that venture on the dorsolateral migration pathway by chemotactic factors and cell matrix glycoproteins. The first neural crest cell to migrate in the chick entered the ventral pathway followed by the dorsal pathway. Cells migrating dorsolaterally remain above the neural tube become specified as a melanoblast. FOXD3 transcription factor controls the switch between glial and neural and the melanoblast precursor. In the presence of FOXD3, the transcription factor necessary for melanoblast specification and pigment production, MITF, is repressed. When MITF is downregulated, then the cells become melanoblasts. MITF is involved in three signal cascades. The first cascade activates genes responsible for pigment production. During the second cascade, neural crest cells travel along the dorsolateral pathway into the skin. Third, cascade prevents apoptosis in migrating cells. The heterozygosity for MITF can cause there to be fewer pigment cells in the center of the body and the middle of the hair. In mammals, the KIT receptor protein is needed in causing committing melanoblasts to migrate on the dorsolateral pathway. KIT is located on neural crests that also express MITF, which are presumptive neuroblasts. KIT binds to stem cell factors made by dermal cells. KIT bound to SCF, or stem cell factors, prevent apoptosis and stimulate cell division of melanoblast precursors. If there is an insufficient amount of KIT, neural crest cells cannot proliferate efficiently over the entire skin. In the feather or hair follicle, melanoblast becomes melanocyte stem cells. In vertebrates, melanoblasts enter the epidermis into the hair follicles or feather primordia and pollen to the base of the follicle. In mice, all of the melanoblasts go into the hair follicle, while in birds and humans, the epidermis and cutaneous appendage become pigmented. The melanocyte stem cells are supported by the hair follicle stem cells, which allow the cell membrane to be made of collagen. This is needed for self-renewal of hair follicle and melanocyte cells. Melanocytes migrate across the border of the hair follicle in the beginning of hair development to differentiate into mature melanocytes and provide pigmentation to the hair shaft. Epithelial cells secrete FGF2, which stimulates melanocytes to transfer pigment into the epithelial cells. This is the reason why mice and humans' hair turn gray as they age, because the melanocyte stem cells deplete. In the feather or hair follicle, melanoblast becomes melanocyte stem cells. In vertebrates, melanoblasts enter the epidermis into the hair follicles or feather primordia and pollen to the base of the follicle. In mice, all of the melanoblasts go into the hair follicle, while in birds and humans, the epidermis and cutaneous appendage become pigmented. The melanocyte stem cells are supported by the hair follicle stem cells, which allow the cell membrane to be made of collagen. This is needed for self-renewal of hair follicle and melanocyte cells. Melanocytes migrate across the border of the hair follicle in the beginning of hair development to differentiate into mature melanocytes and provide pigmentation to the hair shaft. Epithelial cells secrete FGF2, which stimulates melanocytes to transfer pigment into the epithelial cells. This is the reason why mice and humans' hair turn gray as they age, because the melanocyte stem cells deplete. Now that we have discussed what exactly neural crest cells are and how it relates to skin pigmentation, it is now time for us to come back and take home key points. So one thing you must know, neural crest cells can migrate to become different types of cells: cranial, cardiac, trunk, and the vagosacral. Also, the path of neural crest cells depends highly on the extracellular matrix or environment that the neural crest cell migrates to. In trunk neural crest cells, migration can occur in two different ways: dorsolaterally or ventrally. And lastly, melanocytes migrate across hair follicle in the beginning of the hair development cycle, and then they differentiate into mature melanocytes to provide pigmentation to the hair shaft.